Hi, welcome back. My name is Jayanti Mukherjee, and today, as a part of I Think Biology NPTEL course, we are going to do a tutorial on how to measure biodiversity. So, I hope from the lecture you remember that there are different levels of biodiversity that we talked about: species, genetic, ecosystem diversity, and functional diversity. And then we also talked about how to measure species diversity. You can use various indices and also various other comparative methods have been done. But in this tutorial, we are just going to do a basic measurement of biodiversity with Shannon-Weiner index. Okay, so now you actually pause this video and go to your Excel, open an Excel sheet, and we will do the tutorial there. As you can see, I have opened this Excel sheet and I have here site 1. So you should actually pause this video again and copy this full table that I have made first. Okay, site 1, you can use species A, B, C, D, E, F, G and use the second column is abundance or the number of each species. So for example, if you go to an ecosystem or an area, you will see there are 100 mango trees or 4 jackfruit trees. So that is the abundance, right? The count of it. So this goes into the second column. And then I have put PI, which is, if you remember the formula that we talked about for species diversity, PI is the proportion of species A in that ecosystem. Okay, we'll take just a minute, we will see how to do that. And then ln, or natural log of this proportion. And in the third column, if you remember the formula, it will be pi, the proportion, times the natural log of the proportion. So if this you make the table first, then we will start on how to do it. Okay, so let's get started. We will count the total abundance of this area. Okay, now let's get started. We will see how to get the sum of these or the abundance, total abundance of the area. So for that, put your cursor on this cell, add an equal sign, write sum, and then a bracket, and you then select all these cells, right? I'm sure you have already done these kind of calculations and you know a lot about it, but this is just for our convenience now. Then how to get a proportion, PI or the proportion, we again select this and then go to put the equal sign and put a bracket, select this cell, okay, and then you can also do two ways. You can do it. One, divided by, you can select this cell, okay, and then you can get this, or and I can draw this again, So see, this is the reason it may not give you the result. Okay, so the next another way that you can do it is you select this, select the number or the species abundance, you can put this and just type the number. That is the easiest way. Okay, you can type the number, that is the easiest way. There's another way you can do where you can actually fix this cell. There you can use the dollar sign to fix that cell, but we are not going to do that today. Okay, you type the cell and then you get these values. So these are the proportions of each of the species in this area or ecosystem. Then, as you can see, based on the number of the individuals, the proportion of A is much higher than most of them, and this G is the lowest now. Then we go to the second column. You can type another equals and then select LN. So ln is the natural log. Of course, if you type just log, you will get different values. But the formula says ln, so we are going to go with the formula and we'll do ln of the number. So this is the number because it is ln pi, right? So we'll select that. And of course, as you know, all the log values will be negative. So you can actually get this number. In the third column, as you see, what do we have to do here? This is multiplication, right? This is multiplication. And this, let me put a, yeah. So, to do the multiplication, I select this first column, Pi, and then I do a multiplication sign, and I select this, and I then put enter. So, this is my final value, right, of the third column. Now, if I go to the formula back, you will see Shannon-Weiner index is the negative of the summation of Pi times ln Pi. Okay, negative of the summation of Pi times ln Pi. So that is what we are going to do here. Equals negative sign summation of these values of Pi and ln Pi. Okay, and you close both the brackets. So as you can see, first the red one and then the black one. So both the parentheses or bracket has to be closed. Only then you will get your number. So this is the Shannon-Weiner index. Okay, I will write here total abundance. This is the index. What you can do is cut from here and you can paste it here. So this is your Shannon-Weiner index. Now, why have I given other columns? As you can see, the next column here, why it is there. So just to take you through this, you can see in the second side, which I have written here, also you have A, B, C, D, E, F, G, but now the difference you see is in the number of individuals that are present in that site. So here the individuals are more equally distributed. Okay, so the abundance is very equal, and that is the reason we talked also about another term which was evenness. Okay, so the richness of this site A is, if you select this, you will see it will be 7. 1, 2, 3, 4, 5, 6, 7, right? 7 species. Here too you have 7 species and all the species are same. But the abundance is now different in both the sides. Here the abundance was highly unequal. One was just one individual, one was 5, whereas some was almost 300 individuals. So now here the abundance is more or less equal. So now let's see whether with higher evenness in site C, whether we are finding the diversity is high or not. Okay. So we will do the same exercise here. Equals, sorry, not here. First we will get total abundance, right? And then you will get equals sum. The same procedure you will do. This is the sum. Then Pi first. I will just insert the number here rather than the formula now for far or ease of use. Okay. Second will be ln pi. So ln log of pi. Natural log of the proportion. Second is the gain. Sorry, let me just copy this here. Okay. And now here again, this times this. So as you can see, I'll just tell you, as you can see here, I use the bracket. You can see I use the bracket, and here probably I did not use it. So when there are only two numbers, you don't need to use the bracket. But if there are multiple things in one line, for example, one multiplication, one division, one addition, something like that, then definitely you should delineate them with separate brackets. Okay, otherwise your formula will not work. So this is something to keep in mind here. Now let's do the same thing. The formula which was negative, you can see negative of the sum of this column. Okay, so equals to negative sum of this column. I have to put another bracket here. If not, I can, or okay. So then because it is negative, now negative negative cancel, and now it becomes a positive. But the thing to note, the most important thing to note here is, you see, if the evenness now has increased in this site, then the diversity also increases. So a measure of diversity, to estimate diversity, both evenness and richness has to be taken into consideration. Because just richness, say somebody gives me to do an environmental assessment, to go to two different sites and I just go and measure the richness and come. Whereas in one area, all the species are equally distributed, which means that their population in that region are not that vulnerable. I won't know. If I don't measure the evenness, I won't know what is their population status or density status rather, right? But if site A, if I go and one species has just one individual, and if I don't measure the evenness or the abundance, then I won't know what the area, how threatened one species is, or how the diversity is actually. Okay, so evenness actually is very important to measure. So that's the reason this index actually combines these both parameters, right? So now let's go to the third side. Third side. So here you see, on the first side there is 1.5, right? Diversity is 1.5 approximate, and this is 1.9594. Now let's see what happens if we now increase the richness. So now the number of species has also increased, but again the evenness or the abundance evenness is very variable. Okay, so now let's do the sum. Okay. 612. Now we go here, this divided by 600, got this number. Same I will do ln of these, okay. Now you should also keep doing simultaneously. So now we will do the multiplication of this times this. Okay. Now let's see the summation. Okay. So now it is, you can see it has increased much more than the first one, because now the evenness is, although it's very uneven still here, but the evenness is still little bit equally distributed, and the variation is not this much from one individual to 300 individual. Okay, so here evenness was it was not even at all, right? But here when you come in this third side, there is a lot of variation, but still it is in a manageable level. So the diversity is 2.13 now because of the increase in richness. And the fourth site I have included, again I have included the evenness, very even. So we should now hypothesize, right, that the last or the fourth site diversity would be the highest. Okay, that will be our hypothesis. Now let's see if that is the case. Sum again. Okay, now it's 1639 individuals pi divided by 1639 individuals. Okay, now natural log of this proportion gives us this. Now pi times lnp. Okay. So according to a hypothesis, now let's see, putting the formula, what happens. Negative sum of all these, right? So definitely you can see that there's a significant increase in the diversity from the third side, because now the richness although was similar, but again the evenness changed, or it became much more even. The side became much more even. So this is a very basic and simplistic way of understanding how to measure diversity. And as a preliminary analysis, you can always go to different sites. Say for example, you can go to the backyard and you can collect some plant species or something which you want to measure the diversity of. You can do this small exercise at in your backyard or somewhere with your friends for fun, and see, compare their backyard with your backyard to see which backyard or which garden is more diverse. Okay, so have some fun. Now we are in the next sheet. Here you see I have come to beta diversity calculations. As like the previous sheet here where we did, 4 different sites calculation. Similarly, I have 4 sites here. But you will see how I have made the differences of showing you the 2 components of beta diversity that we talked about. One was species nestedness and species turnover. Okay, so the similarity as well as the dissimilarity. So in site 1, you can see I have given 1, 2, 3, A, B, C, D, E, F, L. Okay, now site 2 has A, B, C, D, E, now H, I, J, K. Okay, so the A, B, C, D, E are common to both the sites. Okay, which makes 5 species common to both the sites. So if you remember the formula, what we talked about, beta Sorensen was 2A, which is A was the common species between the 2 sites. B was common unique species for say site A, and site C is the unique species for site B or site 2. So if we have A as 5, okay, we will have B, which is say unique species for site 1, which will be just 1, okay, and unique species for site 2 will be C, which is 1, 2, 3, 4. 4. So you can see that site 2 also has 2 extra species, number of species. Okay, so richness anyway in site 2 is greater. But let's see how the similarity or turnover is. What is the measure? So we will incorporate the formula here. 2, you will put a bracket, 2 multiplication of 5. It's actually simpler to do this way than to select the cells, because if you select the cells you have to put $sign, and also it gets a little bit complicated. You can do it this way or either way. Divided by, again one bracket, you can put again 2 multiplied by 5 plus 1 plus 4. Now you can actually give another bracket here and make it complete. Okay. So the beta Sorensen or similarity is 0.666, which is say approximately 67%. And it is very clear from this site also that similarity in these sites is quite high. And this similarity will be then is equal to 1 minus the similarity, which is of course 0.33 or something like that. So now let's do the site 3 and site 4. Okay. So here I see A, B, C, D, E, F, G, and here you will see, so 7 species. So richness is 7. On the other hand, site 4 has richness 12. So quite high richness. Now let's see how it is in terms of nestedness and turnover. So from the first two sites we saw that this will be actually a measure of nestedness, which is similarities, which is quite high or 66%. However, researchers have actually shown different measures of nestedness as well as turnover. So not to confuse with this, but this is just to simply put beta Sorensen's similarity index. So here we have A, B, C, D, E, F, G. Okay. And so all 5 here, A again, all 7 are unique to the site. Sorry, it will be 6. One C is repeated here, you can see. A, B, C, D, E, F, G. So one C is repeated. So my B will be 6 species, unique species. And A will be just 1. And C will be here 12 minus 1, which is 11. So again I will put the formula. 2 times 1 divided by 2 again times 1 plus 6 plus 11. Yeah, so as you can see, it's just 10% of similarity or nestedness between these two sites. And if you do similarly 1 minus this, you will get much high turnover. So the uniqueness of the next site is compared to the first site is much more. So these two sites are not very comparable. So I think I skipped one part in the lecture section, but I will tell it here. So this calculation becomes very important when we actually look at conservation or evaluate different sites to compare biodiversity. For example, if I go to an area and I want to do, I want to see if how much is the heterogeneity in that area. So by that what I mean is how much does species diversity change with space. So I put say 10 plots and I measure or calculate beta diversity from one plot to another. Okay, and I can actually compare what is the turnover or how many unique species are being added to these plots. So that is a good way of comparing. You can also do it from two big sites. Say for example, I'm comparing Rajaji National Park to Corbett National Park. These two big parks, I'm comparing how much is the similarity in species composition compared to nestedness in species composition. So these are the values of using this index. And there are several other index available for the same purposes, but with some unique attributes to them. I hope you enjoyed this session tutorial on how to measure diversity. And just to tell you a bit more, that in our fungi chapter, the kingdom of fungi, you can see another analysis that we have used to estimate species richness, which is called rarefaction analysis. So this is the uh, book chapter. You can see it's called rarefaction analysis, and it has been step by step. We have given the process and help to understand how you can use just species richness to estimate diversity of an area, and also how you can compare different areas of for their diversity. So if you go through this chapter, if you have any questions, we can always come back to the live sessions and interact with you if you have any questions. Okay. That will be all for today. Thank you very much.